So I was going to actually start my talk by telling this little story. So while he's putting it up, I'll tell you that story anyway. The rabbit died was actually a way of saying that you were pregnant in the US about 70 years ago. And the reason that is actually there's something interesting behind it. What they used to do about 70 years ago, before they had tests for pregnancy hormone, was they used to take urine from a woman that was likely pregnant and then inject it into a rabbit and then observe if the ovaries of the rabbit actually matured. And it was believed, actually mistakenly, that the rabbit died in the process. Actually, all rabbits had to be dissected in order to do this procedure. So they all died anyway. But so it became, in fact, this is a commonly used term in the US, apparently in the 1940s and 50s, that did the rabbit die? And if the answer was yes, then you were pregnant. And thankfully, we don't have to kill any rabbits anymore to determine if people are pregnant, because diagnostic testing has come a very long way since then. So that's really going to be the subject of my talk today, is, just in time, blood on a chip. And in fact, not just blood, but other body fluids, urine, etc., broadly lumped into this category, right? So, thanks. So what's actually in blood? I guess we should start there, because if we're doing testing in blood, presumably there's something important inside it. And I think many of you are already science students, kind of know this, but I'll just repeat some of the key points here. That blood is composed of a cellular component, which is red because red blood cells are red. And then you have the plasma, the kind of clearer component, which carries a lot of water basically, but also a lot of proteins and other nutrients. And even within the cellular constituents of blood, you have many, many different varieties, especially of white blood cells. There are several different categories of these leukocytes. And then, of course, you have the largest volume really is of these erythrocytes, or the red blood cells that carry oxygen around your blood. And then you have a small but important number of platelets also in your blood. So that's what's really inside blood, okay? So what does blood really do in the body, right? And that kind of determines why you need to test blood also. So there's very broadly 3 different things that blood does. It transports, which is kind of obvious. It's moving around your body. It protects us from infections and actually helps regulate your physiological balance. And, no, I'll stick there, yeah. So without going through all of those, I'll just talk about the ones in red, right? Because we don't have time to go through everything. But each of the lines that I've got in red are kind of talking about important physiological imbalances potentially, and tests that actually are used to diagnose that stage. So diabetes, again, a very well-known phenomenon, unfortunately, and glucose tests that actually, when done on fasting patients, you could actually pick up whether they're diabetic or not. And really, the reason you do that from blood, of course, is because blood is transporting glucose around your body. Similarly, kidney failure, the ratio of urea creatinine tells nephrologists what the state of your kidney is, whether it's healthy or not. And again, that's because blood is actually transporting toxic waste out of your body. Even in cancer, in the unfortunate event of a metastatic tumor, you might actually have a cancer cell that gets transported through your blood. And today, called liquid biopsies, they represent a very exciting new form of diagnostic tests that can actually tell you before a tumor becomes full-blown metastatic. And in terms of protecting, obviously, the blood does a lot of things by carrying the different kinds of white blood cells and even antibodies that destroy pathogens. So, for instance, a slightly older school version of a lot of infectious disease tests actually measure for antibodies in your blood, because your blood is carrying them around and uses them to integrate or destroy pathogens. Now, at the root of every blood test there's a fairly simple set of things that goes on, right? There's many, many varieties of ways of doing it, but any sensing mechanism, anything that you're trying to quantify an analyte from, is doing something quite simple usually. So you have 3 classes here shown in yellow, blue, and sort of the light yellow there. So you first have some sort of a receptor. The receptor could be an antibody, it could be an enzyme, it could be another cell, for example. So in the case of, I'll take glucose as an example here. You have an enzyme, glucose oxidase, which actually specifically recognizes glucose. And by that action, then sets off a series of reactions which in the most common case of glucose sensors, it's actually sitting on an electrode. You have some electroactive materials there, and that generates finally a current. That's the entire signal transduction part. And then that goes to some signal conditioning circuitry, some electronics, and that gets spit out finally as your glucose value. So pretty much every diagnostic test that's done has some thing within here. I just went through one example for glucose, but you could imagine a similar example with light transduction instead of electronic transduction, for example, or piezo, that is mass change transduction, and so on. But fundamentally, you have these three pieces of any diagnostic test. And by the way, I hope this is not over simple for all of you, because if everybody is an expert at this, it's a bit of a waste of time. Does everybody already know this? Should I skip things like this? Okay, good. Somebody said, one person said, go through it, so I'm doing it. So what happens in a lab, right? And we're going to talk about blood testing on a chip or point-of-care testing. But what happens in the traditional large diagnostic lab? So many of you have probably seen ELISA or microwell plates. So very often you have some form of your test happening in a plate system. And you can see there that gradation of blue from light blue to very dark blue. And that's usually from some calibration curve, for example, that you're running there, where you're increasing the concentration of glucose, say, and that becomes proportional to some colorimetric signal, and that blue is then related back to glucose, right? So the microplate actually is sort of, you could imagine in a simplistic way, is what happens in a large lab. You have hundreds of samples, they're well barcoded and so on, and you have all these automated liquid handling systems moving around, pipetting things into one well, mixing things, reading a colorimetric product, moving things around, etc., and finally measuring them using a spectrometer. So that's typically what happens in a large lab setting. The other thing that's important to mention about tests is that they come in a variety of categories. So this is a breakup of the Chinese market. You have, as you can see, 1, 2, 3, 4, 5 different colors. In fact, there's probably a few more categories. But you have broadly immunoassays, which use antibody-antigen binding reactions. You have a lot of chemistry, in fact, that's clubbed into immunochemistry, which are usually enzymatic assays converted finally into some color or electric signal. Then you have tissue diagnostics. So, this is immunohistopathology, and these are actually imaging kind of measurements. You have molecular diagnostics, which use things like PCR or today next-gen sequencing to actually probe directly for DNA, RNA. And then you have this broad category of POCT and SMBG. Does anyone know what SMBG is, by the way? Anyone wants to take a guess? That one test alone is 16.6% of the entire diagnostics market. Can you guess what it must be? One single test. Yes, it's blood glucose, right? It's self-monitored blood glucose, so SMBG. It's kind of awkward acronym, but this is like the Roche AccuCheck and all of these systems. So blood glucose, by virtue of the fact that it has gone all the way to the patient, is actually an enormous test measured in dollar volumes, right? And that alone dwarfs any other test that's available on the market because it's gone to the final patient. So let's also look at the scenario in terms of diagnostics today. And this is broadly India, but in many other countries you have the same. So you usually have some tree-like structure. On top of the tree, you have these reference labs. So for India, you have, for instance, Thyrocare or Metropolis, SRL. These are all the big reference labs that run large facilities with highly automated equipment that, with very few people, is able to do an enormous number of samples every day, correct? And these labs are usually stocking kind of large automated equipment from Roche and Abbott, Siemens, big companies like this. But if you look at where those samples originate, many of them come from doctors' offices, small labs, collection centers, even from people's homes today, right? So those samples actually find their way up this tree into these reference labs. And a lot of testing in the past, of course, has been built around heavy automation and building for the reference lab. But what we're going to talk about a little more today is more focused on these collection centers and doctors' offices and so on. So why is it actually important to even be doing this testing at these POC settings? Because one could argue, in fact, that these large labs get very low-cost reagents because of economies of scale. They have good quality processes. They have a lot of automation. So why can't we just do all testing there? So these are some of the reasons why that's there's a move away from that. And I'll just talk about the 3 ones that I've highlighted in the circles. One of the important reasons, especially in Indian context, is actually lower patient compliance. So TB is often given as an example where every patient that carries TB could infect 15 more people per year. And so if you actually send someone off to do a test and they don't get it done, which happens, by the way, very often in India, that person is a healthcare risk to their entire community. And you would like to, in one loop, one visit, actually do the diagnostic test and get them on medication. So that's a good scenario, a good example of where a point-of-care test is actually useful. The second is inaccurate results. So you don't have very good cold chain many times in India. So samples degrade by the time they reach the central lab. So there's a case to be made that you might not actually be reporting accurate results. And so you might be better off, especially in more remote locations, doing it right there. And we all know that we have a vast, untested population in our public health centers that need to get more access to testing. So a little bit of a very brief history of point-of-care diagnostics, since I've talked about why it's important. Starting with uroscopy. So this is something that is basically the study of urine. It's obviously out of fashion right now, but for hundreds or thousands of years, physicians actually examined urine. They smelt it, they even tasted it sometimes to know and diagnose what was wrong with people. So a very classical example that's given is of actually sugar in urine, and ants actually getting attracted to somebody's urine is a sign of diabetes. And so people had actually ways of doing diagnosis like this quite crudely from urine. And in some ways, some of these are making a comeback because urine is a material that's very easily available, right? And much more easy to get than blood. But if you talk about true point of care, the rabbit ride that I talked about, that was when you could say point of care testing really started, 1950 or so. In fact, the first test that really came out is this Ames glucose strips. So there was a company, a pharmaceutical company, that popularized this particular combination of a glucose strips and a reflectance meter. And you can still see, obviously, this is an era when we didn't have LCD screens. So that screen is like an old multimeter or something, right? So people read their glucose on things like that about 40, 50 years ago. And at the same time, you actually had the home pregnancy test. In fact, made by a product designer who was a woman. She was not a biologist or a biochemist. She just felt the need to have a home pregnancy test for women to be private, and she actually came up with this concept, patented it, and so on. So all of this happened in the late 1960s. And then slowly, I'd say there's been a lot of developments. Today, you have the Clear Blue. It's a digital home pregnancy test. It's very expensive because you're really disposing something which has an LCD screen inside it. But in the US, I think it's quite popular right now. Then you have the Abbott iStat, a very, very commercially successful point-of-care analyzer for blood gases and electrolytes. The Agomatrix is a very interesting one. That was the first diagnostic test that actually got connected to an iPhone. So this is, if you see, this is a fairly old iPhone, iPhone 3, I believe. And that was a glucometer that plugged directly into the iPhone at the bottom of the screen, you can see. And strangely, it was not a mass hit. One would have imagined that at the time that iPhone really took off, this thing would also take off. But what happened was, and this is where you realize healthcare is so different than consumer electronics, that every new generation of the iPhone that came out, they had to separately go through regulation all over again for their glucometer. So that kind of cost was probably not worth it because you're not selling these things in the hundreds of millions like you are with other consumer electronics. But again, these kind of things are making a comeback right now, I would say, commercially. And finally, going all the way to sort of microfluidics, lab on a chip, lab on a CD there. And today, we're even talking about doing sequencing on something that's connected to your laptop with Oxford Nanopore. So these are all just some kind of examples and a snapshot of going from uroscopy to the most advanced sort of point-of-care testing available today. So what does the word chip mean to you? I think for most people chip, because it says blood on a chip. So I was trying to say, why do we say chip, right? Yeah, this is also chip for people maybe who are, I don't know, the wood industry, so wood chips. And then you have semiconductor chips, which is where really the name comes from. And ICs, actually the integrated circuits, because it's a small piece of silicon wafer, they got called chips. It's just a sort of very colloquial way of calling them. There's no particular reason why. So that name has kind of stuck. And now you have microfluidic chips, which are basically, instead of carrying electrons, they're carrying body fluids. So this is a cartridge that we are working on, for example. that is able to carry different body fluids and does some simple molecular tests. Okay, so if you take nothing away from this talk, just remember these three things. What does microfluidics do? Or what does this lab on a chip stuff do anyway? It does three things: miniaturize, integrate, and automate, right? So whatever is happening in the lab, so here's a picture of a big lab. In fact, this is Thyrocare in Mumbai, a large automated lab with literally kilometers of conveyor belts that are carrying around blood samples from one place to the other. So you want to miniaturize all this, right? We don't have that kind of room everywhere. You want to integrate. So this is very, very important, by the way. Integrate means, and that's why the IC became such a big deal. The integrated circuit was not a bunch of disparate things on a breadboard, right? It brought together all sorts of circuitry on one tiny chip. So you want to do the same thing with fluid processing. You want to integrate, for example, centrifugation or plasma separation. You want to integrate some mixing. You want to integrate some elution. You want to integrate some final detection. All of these things which are done separately as different manual steps, you want to integrate onto a cartridge. And finally, you want to automate it. You don't want any manual intervention. You'd like everything to be at the press of a screen. So that's a microfluidic chip at the other end, where you can see sort of the same miniaturize, integrate, automate kind of concept, where somebody's putting in a drop of blood. They have a bunch of reagents that are actually on the cartridge already. They have some dried reagents. Then they have some filters. So you're doing some splitting. You're doing some final, of course, sensor readout. You're doing some mixing. All of these things happening on one card. So that's really the lab-on-chip paradigm. And when has this become really popular, or how has this field evolved? It kind of started with the semiconductor revolution, actually, and that started in the 1950s, as many of you know. So people started making smaller and smaller devices in silicon, and then around the late 70s, early 80s, people said, why can't we apply some of the same miniaturization to analytical chemistry? So people started building things like miniaturized gas chromatography, for instance. And so, but that was still very expensive. It was happening in glass and silicon, and it's kind of very expensive when done on a small scale. But then in the 90s, there was something called soft lithography that got invented. So if any of you have played with PDMS, which is a very popular polymer in the field of microfluidics, you'll know that that's very easy to work with, and you need a master wafer from which you can make lots of these microfluidic chips. So that really, I would say, caused a mini explosion in the field and in the number of publications and so on. So now, really making miniature features, and when we say miniature, we're talking about 100 microns. So this is not miniature by semiconductor standards, where today people are like at 10 nanometers, right? So we're quite big. But when you look at physiology and cells and so on, you don't want things that are that tiny. So we're at about 100 microns typically for a microfluidic device. But being able to reproducibly make features like that, even in plastic, is not a trivial challenge. So some of those engineering problems have got solved over the last 10 to 15 years that has really led to the rise of this field. So how do you make these microfluidic cartridges? So this is a very common way to do it in academia, where you actually have a master, and then that contains these micro features. You pour a polymer, you cure it, and that actually contains these features which you can then peel off, and you can do this process repeatedly and use it again and again. You can also do CNC micromachining with a very, very small tools. But really, from an industrial point of view, the most favored process is injection molding. Injection molding is how lots of commercial grade plastics are made today, bottles and anything plastic that you see around you is typically made by injection molding. So this actually is sort of a challenging injection molding because you have very fine features down to about 0.1 millimeters. Most injection molders are comfortable around the 1 millimeter type of length scale. So if you do micro molding though, you can make lots of these parts and you can make them very cheaply. So that means you can make a microfluidic chip or a cartridge or a cassette cheap enough that it can be disposable. So this is the key point because you're dealing with blood. You can't reuse these things. So you want them to be disposable because of that reason, and that means they also have to be quite cheap. So here's this example of a very early device, a microfluidic device, that was actually by one of the pioneers in the field, Steve Quake, and he's actually at Stanford. And he came up with this whole concept of valves in microfluidic devices, Quake valves. And this simple device, what it was doing was actually taking bacteria, lysing it, that is breaking it up and extracting the DNA from it. So if you think about it, it's fairly simple. In fact, some experienced practitioner in the lab would do it quite quickly as a bunch of steps. But something like this, even though it seems simple, is not that easy to automate. Because everyone wants today things in what's called a sample in, answer out format, right? I want to put in a drop of blood or put in a drop of saliva, and everything after that has to get automated. So what this device does actually takes lysis buffer, dilution buffer, shown there in yellow and green, and then each of those things that are crossed out are valves. So by opening and closing these valves and using sort of a peristaltic concept, he actually mixes those three buffers, lyses the cells, and then the cells flow further downstream where the lysed cells now, this DNA is out of the cells, so they bind to a column, and then you have another elution buffer that actually removes the DNA off of that column and then sends it to a separate waste channel or separate channel for further use. So a very simple but important set of steps that got automated on this microfluidic chip, and you can see that's a US dime, so like an old 25 paise coin, but probably many of you have never even seen those, so it's quite small. And what's happened over the last 10 years is people made some jokes, by the way, about is it lab on a chip or is it a chip in a lab? So because a lot of things look like this, that the cartridges were very small, but they have tons of tubing and pumping and all sorts of expensive setups that went around with them. But now the field has evolved to the point where this is kind of a latest generation microfluidic chip from an Australian company called Minifab. It has integrated reagents, it's very nicely designed, and I think it's a final electrochemical readout, so you can see the electrodes there. But it's a fully integrated chip that just plugs into a reader and does a fairly sophisticated set of analyses. So it's not just about making the chip. When you're trying to make a product, it's about getting all of these things right. Adapters, interfaces, bonding, reagent loading, all of these are equally important. I'll come back to that in a second. Very briefly, I didn't introduce myself. I'm Dhananjay, and I run this company, Achira Labs. So we are actually a company that is trying to do a combination of both innovative engineering as well as commercialize this product. We're building point-of-care immunoassays, so one of the verticals that I talked about earlier. So this is our product, and I was asked to give, by the way, as general a presentation as possible and not sort of do a marketing pitch for my company, so I'm trying to do that. I will tell you very briefly about our product. So this is, like a lot of microfluidic lab-on-chip products, is a combination of an instrument and a plastic disposable. So that's our disposable, and it's that micro-injection molded piece of plastic that I talked about. And the concept of it is actually very simple. So we have these hydrogel biosensors that we actually make ourselves, and I personally have done a lot of work on actually making hydrogel biosensors for several different analytes, including antibodies, oligonucleotides, and so on. We make these sensors in large numbers, and then we embed them inside a microfluidic cartridge. So you have shown there red, green, and orange to do, for example, 3 different tests. And it's a cartoon, so you could do this 20 or 40 or 50 different tests even. And then you flow through this cartridge a very small volume of sample. So one of the challenges, by the way, with the traditional lab analyzers is they need a minimum amount of blood, which is about 0.2 milliliters of, not blood, but serum, which is the clear component of blood, to even begin any process. So that's 0.2 ml of blood. If any of you have done any experiments, you know that you can't easily get that from a person's finger stick. You only can get it from their vein, right? And that needs somebody called a phlebotomist, and a phlebotomist is sort of a specialized person, and you can't get this in a doctor's office context. So that's why the microfluidics has become important here for the miniaturization reason that I said earlier, that you have a very small volume of blood, more like 0.02 ml, an order of magnitude less, but you still want to get all that information from it. So can you do that? So that 0.02 ml of blood, basically 20 microliters, flows through here, and we do some simple binding immunofluorescence chemistry that generates a fluorescence readout, and we actually use that to report the values of hormones on our platform. So we've done several different hormones, and we've validated against gold standard technologies to prove that we get very accurate results compared to them. I will spare all the details. This is us going through a clinical trial where we've placed our analyzers and actually performed all of these tests in triplicate. So that's one platform that we have. And we're building a bunch of hormone, vitamin, and other tests on it, all for doctor's office. And the next version of our reader is a much more smaller, miniaturized version of it. But the other thing that I wanted to mention is, since we talked about chips, we have something else that we worked on at Achira. It's called Fab Chips. It's supported by Grand Challenges Canada. So people have worked on paper, by the way, as a substrate to do also these sorts of lab-on-chip tests. And we said several years ago now, how about trying to do this on yarn, on fabric, right? Basically another capillary medium. So it's a very simple concept where you take yarn, we started with silk yarn, and then you coat yarn with different materials. It could be enzymes or any other oligos or whatever, and then you just weave it on a loom, and you create a patch of fabric that you can then cut into individual test strips. So for instance, this is something that we've done to make low-cost glucose test strips using a loom. So this is a simple hand loom that you can make these on and make very large numbers of them. And in addition to actually making these, you can pattern the silk. So you can have hydrophilic and hydrophobic parts of the silk. So this white stripe here is actually hydrophilic. So when you put a drop of blood, it only wicks along that. So you get some sort of patterning effect to do these kinds of things. Okay, so how many of you have not heard of this company? Oh, actually, quite a few. I see, that's interesting. So I thought I will address this question because everybody in this field gets asked about this company. And I will spare you too many details. You should go read about it later online.
That's a very interesting book.
And there's an interesting book. Yes, you can buy that book. That's why Bad Blood. So in short, Elizabeth Holmes is an entrepreneur in the US who raised a lot of money and a lot of hype about doing point-of-care tests with a finger stick of blood, and got $9 billion valuation, raised $700 million, and so on and so forth. But it all fell apart very spectacularly about 1.5 years ago, because a lot of the claims were found to be not true, and et cetera, et cetera. So go read about it separately. I just thought I'll address it because this field of point-of-care testing, everybody who's in this field gets asked this question, that how do we know you are not Theranos? And unfortunately, sometimes when you have a leader who can actually lead the field, but here the leader actually kind of had made life difficult for everybody else who's trying to raise money. But just 2 or 3 points about learnings from the Theranos story. I think first of all, we have to understand that they're all still in a field that's quite close to academic science. So really convincing people that whatever you're doing works means doing peer-reviewed publications, working with clinicians, exposing your technology and not being in stealth mode and hiding it. These are very, very important because it's healthcare and people need to be convinced that whatever you're doing is kosher. Secondly, very important to know that tech development is very time-consuming in this space. So the Silicon Valley model of, move fast and break everything or whatever it is doesn't work here. Because if you did that, you might just have a spectacular failure and you're playing with people's lives at that point. So you can't do that. Things need some time to get validated, tested, manufactured, regulated, et cetera, et cetera. And the other thing is to not be too ambitious about what you're doing. So for instance, we are doing a small number of tests and then we hope to build up to a large number of them. So that's just some of my own personal learnings looking at what's happened with this company. So I will, I'm coming to, how are we doing on time, by the way? We're okay on time, okay. So I want to talk a little bit now because all of you, many of you students here, what is sort of the future here, or what are some of the interesting trends that are happening? So one of the most promising ones is really actually telemedicine and what's happening on our phones, right? I don't know if any of you do these consultations on the phone today, but they're becoming more and more popular with many people, that you do a consultation, and there are several companies now in India even offering basic consultations on the phone. And you could imagine that when tied up with that, diagnostics takes on a particularly important value, correct? Because if doctor's not able to see you directly, they're only seeing you through the phone screen. But if they could, for example, say, hey, why don't you also, can you get just this tested almost immediately in real time? And if you could give that number back to them and they could actually then, based on that, say something, that would be quite an interesting and exciting world, especially for, let's say you have a pediatric infection or, a young child with a high fever, the parents are scared, it's the middle of the night, they don't know what to do, and they'd like to get some sort of answer about what's happening. So you could imagine several such scenarios where, this could be a very deadly combination and very helpful to people. So there's several gaps though, right? What I just outlined, by the way, is not close to happening right now. And why is that? Obviously, we have smartphones. They're quite cheap. Everybody's got a smartphone. Cloud computing software exists. A bunch of smart sensors also exist. Our phones are full of sensors, accelerometers, and things like that. But what we're really missing are the sensors that deal with body fluids, right? So that thing, exactly what I talked about, if I just said, can I take a drop of saliva or tear fluid or something and just put it into something which magically then creates a signal that can be read, and can that be quite cheap? That's not there yet, right? So there are several open problems in this area which are around sensors for blood testing or body fluid testing. And I've just laid out some of these. I'm not going to read out everything, but some of the important sort of gaps. And not always, one of the mistakes, classical mistakes we always make is we say, oh, I'm building a cheaper version of X, right? And many students, in fact, I spoke to earlier, so many people had ideas about I want to do a product startup, I want to do a company based on an innovation and so on. And so one advice that I would give you is don't start with something already exists, I'm going to make it cheaper. Right? Usually you'll find that if you go down that road, A, it's going to take you several years to get to where that person is. They could probably make it cheaper by the time you actually get there and so on and so forth. So you want to look at real differentiation beyond cost, correct? There's sometimes, by the way, when making things cheaper is useful, but it's not always. So here's just a list of potential things. For example, typhoid, I can tell you, there is this very cheap typhoid test already out there. It's just not very good. So the real problem in that space is finding a good antigen and then making reagents for it, right? So the problem is more on the biochemistry side, biology side, rather than the technology and the test development side. And one of the sort of very important tools here is to make a target product profile of this kind, just a table which says what exactly are you trying to do with your test. So this is something that I just put down for glucose. It's a very common sense thing to say, but it's an important exercise to do if you're trying to develop any diagnostic test. What exactly are all of the different parameters that you hope for your test to hit? And that will actually tell you a lot of things about what's possible and what's not. I'll finish up with a few lessons learned doing this whole microfluidics lab on a chip thing. One is manufacturing. Actually, manufacturing of these cartridges can be quite complex because there's several different child parts. So that's our cartridge, and this is purposely blurred so that you can't replicate what we're doing, okay? But it's got a lot of steps. So that's what I want to convey to you, is that there's many, many things that go into making a cartridge, right? Lots of QCs, lots of assembly steps, lots of checks, et cetera, to make a quality product. And lots of manual operations too, because you can't afford to automate early on. So what that means, this is very important to note, is another mistake people often make is they say, oh, you know, somebody is making the X product and they're selling it for 500 rupees, but if I add up all of the costs, it's not even 4 rupees. So then, you know, I can start doing this tomorrow and I'll do it for, I'll sell it at 100 rupees and I will beat them, right? So one thing that you should remember is the cost of goods is very different from the bill of materials. So when you put something like this together, you'll often find that the cost of assembling this is actually more than just the cost of all the child parts. So a very simple thing, but important when you're doing an economic analysis. The other lesson is many people get into diagnostics thinking from a Mother Teresa-like point of view of the world that I'll make everything cheaper. But unfortunately, as a technology provider, only a few things are in your hand. What you sell it to at the lab is really what you can decide. But beyond that, what you'll often find is if you make things cheaper, people will keep the benefits for themselves and charge the patient the same. So bringing costs down for the patient is very different from bringing down the costs of technology. So there are more complex business model issues involved here that you should be aware of. Lesson 3 is it's very expensive to build a platform, right? The kind of thing that we're doing is both expensive in terms of time and money. So we've got some numbers from the US, unfortunately not so much from India. The average diagnostics platform takes about 7 to 8 years and about $30 million before it gets into the market. So that's not what people imagine. They think that it must be a lot faster and cheaper and only pharmaceuticals are expensive, but that's not true. Diagnostics platform building is reasonably expensive and is not got the same kind of payback as a blockbuster drug. So I'll finish with some important trends, I think, just for especially some of the younger people. Going forward, I think some very exciting areas are wearables, so things that you can wear on your clothes that can do testing, implantable sensors, so people actually putting in a catheter or something that goes into you that's actually continuously monitoring all of the analytes in your bloodstream, so you don't have to keep taking data once in a while. You'll get continuous data, basically, and this is already happening, by the way, in animals. Machine learning, of course, everybody knows is a very big deal, but specifically in diagnostics, I think using machine learning is going to be very important in terms of taking large data sets and correlating it with clinical conditions. You'll need a lot of careful longitudinal studies, so it's not as trivial as I think some people think it is. It's going to need a lot of careful data collection. Another interesting trend is, I think, crowdsourcing. Some people already saw how Google was able to tell when there were asthma attacks in several different parts of the US all at the same time. So in a place like India, you could tell when an H1N1 infection is happening in a certain place. And from a public health point of view, this could be very, very interesting to clamp down early on infections. And early predictive markers for cancer and mental health. I think these are two big open problems that people are very excited about right now. Mental health, telling people that they're going to get Alzheimer's or Parkinson's 5, 10, 15 years from now can be a very big deal in terms of improving the quality of their lives. So this is really a biomarker problem. But identifying biomarkers for cancer and mental health, I think these are both big things. So these are all our partners and funders. So I'll just finish up by thanking all of them and you.